evening, everybody. How are you today? How are you today? Awesome. It's absolutely brilliant to have you here this evening on this warm and wet evening, and an absolute pleasure to have the Australian Embassy with us this evening, who we've partnered on this event with. So we're going to be exploring the ethical issues relating to bionics and stem cell research, and I'm really looking forward to hearing how tonight goes. We're joined, of course, by David Hoey from Trinity College. We're joined by Susan Dodds from the University of New South Wales, and of course Gordon Wallace, who did speak here in 2011, talking about the benefits and challenges of biology being fused with engineering and technology. I'm going to have to dig out the archives and check out that talk. So again, thank you so much for coming tonight. I hope you enjoy it, and we hope to see you again in Science Gallery Dublin. And I'm going to hand you over now to Deputy Ambassador Simon Mamouney, who will be emceeing the night tonight. Thank you so much, and enjoy your evening.
Thank you. Thanks very much, Sarah, and it's a pleasure to sort of be here tonight, and thank you very much for coming along to, I think, what is going to be a thought-provoking and very interesting discussion. The venue here tonight, the Science Gallery, it's a fantastic sort of venue. I'm very sort of pleased to be working with them in hosting this event here tonight. And I was also delighted to learn that there'll be a science gallery opening up in my hometown in Melbourne, Australia in the future. Now, I'm very glad that amongst these very eminent scientists such as Professor Wallace and David Hoey, that the arts is represented by Professor Susan Dodds. I'm a great advocate for the arts, for an arts degree, as it can take you very far. Hopefully I'm an example of that. But I'm still scarred from my university days and from what I read about arts degrees on the bathroom walls of the engineering and science faculties in the University of Melbourne. Now, all of you with science and engineering degrees, don't you dare pretend that you don't know what I'm talking about. But in all seriousness, it's a real honour to be partnering with the Science Gallery with this event. This is exactly the kind of event that we need more of. Australia and Ireland are natural partners. I'd be hard-pressed to find any other country in the world where I would feel more at home and more at ease than here in Ireland. We have a natural bond and kinship that few countries enjoy. With Brexit, we have noticed a welcome focus on this bilateral relationship, as evidenced by a number of high-level visits, including the President of Ireland to Australia last year and our Governor-General to Ireland in September. But high-level visits are few and far between. What's much more important are visits by people like Gordon and Susan here tonight, and they come year after year. Now, for some advertising, why we're here. Recently, the Australian government announced as part of its recent budget the establishment of the Endeavour Leadership Program. This is going to replace the Endeavour Scholarships and Fellowships and Mobility Grants into one program. Now, I mention this program because one of the speakers here tonight, Professor Wallace, has played an influential role in encouraging a number of students here from Ireland to participate. It's a very generous program that offers postgraduate students, professionals, the opportunities to study in Australia for a period of a couple of months up until a whole sort of PhD. Now, when I arrived here in Ireland less than sort of 2 years ago, I was very interested to see, well, how many Irish scholars there were. To date, I've worked out since about 2005, there have been 32 Irish students that have been awarded research, postgraduate and executive scholarship. One of the common threads that stands out more than others is that they are female and that they study in STEM. Now, I'm not sure whether this is because of Professor Gordon Wallace's charming good looks, but quite a number have a lot of links to the University of Wollongong. Sadly, the Embassy's own efforts have been sadly lacking in helping to promote this wonderful program. So for those of you in the audience that are interested or may know someone that may benefit from going to study in Australia for a couple of months, we don't have to keep you. We're more than happy for you to come back to Ireland and develop those links between our two countries, especially in the research areas. I would encourage you to look into this as the 2019 round will open up in June. Now, without sort of any further delay, you don't want to hear anything more from me. I'll now introduce our wonderful speakers tonight, and I'd like to firstly introduce Professor Gordon Wallace. Thank you very much. Is it you first? Sorry? No? It's changed. Sorry, I was reading from the program. So, So, my apologies. So, what we'll do is we'll hear from David Hoey, and then we'll hear from the next speaker, and then the next speaker after that. I can't go wrong there. So, thank you very much, everyone.
Thank you very much, Deputy Ambassador, for the invitation and the introduction. So I'm going to talk to you a little bit about the ethics of stem cell research, but more about stem cell research and a little bit about ethics, because that's really my area of expertise. So I'm a biomedical engineer here in Trinity College Dublin, within the Trinity Centre for Bioengineering. And engineers basically solve problems. And the problem we're trying to solve is trying to find ways to regrow or regenerate damaged tissues that result from injury or from the numerous degenerative diseases that are plaguing our aging population. And so the various PIs within the Trinity Centre for Bioengineering are developing strategies to regenerate all of these various tissues. And the common trend we have in these strategies is that we are using stem cells in each one of these strategies. And so that's why an engineer is standing in front of you talking about stem cell research. So I guess a good place to start is to tell you a little bit what is a stem cell. So a stem cell is an undifferentiated or unspecialized cell that has two tricks. The first trick is that it can self-renew pretty much indefinitely. In other words, you can start with a single cell and result in millions. And the second trick is that if you give it the right cocktail or cues, you can turn that unspecialized cell into a very specialized cell. For example, you can turn it into a bone cell, which can form bone, or a cartilage cell to form cartilage, etc., etc. Generally kind of describing stem cells as two different classes. There's embryonic or iPS cells, which are pluripotent, and we don't do that type of research here in the centre, so I'm not going to discuss them. But what I am going to talk about is the second type, which are these adult or tissue-derived tissue stem cells. And these stem cells are found in pretty much every tissue in every one of your bodies here today. They have enormous potential. It's believed that they can transform medicine because they can regenerate damaged tissues is the hope. And how they are used as therapeutics currently, generally speaking, is that we can extract stem cells from patients, we can expand them on tissue culture plastic, and at that point we can go one of two different ways. The first way is we can put them on materials and give them certain cues that can make them specialized, so we can get them to form bone or cartilage we can then implant into the body. That's tissue engineering. Or we can take those stem cells and inject them into the body, and that is a cell therapy. And what we find is these stem cells home to sites that are injured, and then they start releasing factors that generate a pro-regenerative environment. And so the potential of stem cells are hopefully enormous. And because of that, there are now hundreds of clinical trials using stem cells, or these adult stem cells, to treat numerous diseases, as can be seen here on the bottom left-hand corner of your screen. And interesting, as of last Wednesday, there's 843 clinical trials ongoing around the world. And an interesting thing to point out, if you look at the figure here on your right, the vast majority of those clinical trials are in phase 1 or 2, which means they're only really looking at the safety of these trials with very little impact on the efficacy. So and this trend hasn't changed over the last 10 years. So there's a concern here that these stem cell therapies are not getting through the clinical pipeline, which is a little bit of a concern. And the reasons for this are numerous. There's biological hurdles. For example, all of this type of therapy is relatively new. We don't fully understand how these stem cells work when we inject them in specific locations. We don't know how we should administer them properly. There are regulatory issues. Moving from phase 1 to phase 4 is complicated. It's also enormously expensive. But also, I think it's the fairest thing to say is it's just too early to tell. These clinical trials are still ongoing. There are some very promising results. Some of these have gotten through the clinical pipeline. They are regulated therapies, but we just need to give it some time. The concern here is if you Google stem cell therapies, you will read about these miracle stories about how somebody injected stem cells into a defect and everything is wonderful again. And this isn't helped by looking at professional athletes, which of course the news loves, and talking about Peyton Manning and Rafael Nadal, who have had stem cell treatments to cure ailments. But these aren't part of controlled clinical trials. These are one-offs with no controls. We have no idea if it was because the stem cell was injected or not. And so we have this situation where we have this over expectation of stem cell therapy results in this therapeutic misconception. And that's extremely dangerous because we have thousands, if not hundreds of thousands of patients who are desperately in need of treatments that don't currently exist in a regulated environment. An example here is an article from the New York Times, I think it was Jim Gass. He had a stroke and had no options of treatment. He read about these miracle stem cell therapies and traveled to countries such as Mexico, Argentina, and China, and spent $300,000 getting stem cells injected into him. These worked. He saw actually an improvement in his symptoms over the first 6 months, but then had enormously bad back pain and was unable to walk. And what they found was an enormous mass growing at the base of his spine. And when they removed that, they found that it wasn't his stem cells that were growing, they were the injected stem cells that were in there. And so there is a cautionary tale here, and we need to be aware of this, and these need to be controlled properly. And this isn't just happening in countries that you might be considered unregulated. There's 351 clinics in the US offering stem cell treatments, a lot of which are for ailments that have no clinical evidence that they work. And so this is not in crazy countries, this is in countries that they're in Europe as well. And this needs, from an ethical standpoint, we need to be aware of this and we need to regulate it properly. So for the second half of the talk, I want to talk a little bit more specifically about the research we're doing here in the Trinity Centre for Bioengineering. And particularly trying to understand from more of a fundamental standpoint, how do stem cells behave, how do they work, and what signals regulate this stem cell behavior, and can we combine that with material science and combine it with advanced manufacturing technology to regrow or regenerate whole tissues and whole organs. So in my lab, for example, I'm very much interested in physical cues or physical stimuli and how that regulates stem cell behavior. We all know physical exercise is good for you, right? And it's good for you because when you physically exercise, you physically stimulate the stem cells in your various tissues. Those stem cells sense that and they become activated and start becoming pro-regenerative. But we don't fully understand how all that works. And so my lab builds machines and bioreactors that allow us to simulate that complex mechanical environment in the body, and we can then figure out what stimuli is good or bad for directing stem cell behavior. And importantly, we can also figure out the mechanisms by which stem cells actually sense that physical exercise. And that's important because if we know the mechanisms, we can target them with new drugs that essentially trick the stem cell into think it's being exercised. And that's important because if somebody's suffering from osteoporosis, they have weak bones, you can't tell them to go to the gym. But if we give them this treatment that activates that mechanism, so you can all be sitting there, but your stem cells think you've gone for a run and you start generating more bone. One thing we're also interested in is if we think about stem cell therapies, how does this work? And this goes for nearly all cell research. We take cells from the body, which is a complex three-dimensional environment, and we put it onto two-dimensional hard plastic. The cells are in a completely alien environment. They are not behaving as they behave in the body, and yet this is how the vast majority of research is done. So what we can do is we can develop new fabrication techniques, this is melt electrospinning writing, for example, that allows us to build these three-dimensional fibrous scaffolds, and these fibers are about 50 times thinner than a human hair, and we can completely control their deposition. And by doing this, we can build scaffolds that mimic the real environment that the cells are used to. And by putting stem cells on that alone, we see stem cells look completely different. And when they look completely different, they behave completely different. So we take that 90-degree alignment, for example, those stem cells, that's similar to what we see in bone, those stem cells turn into bone cells and they start making loads of bone. But if you look at the 10-degree alignment, similar to ligaments and tendons, they don't. And so those last two studies I showed you just show how physical stimuli can regulate stem cell behavior. And that's important because when we inject stem cells into the body, we need to know how they're going to behave. And we can use this information to condition cells outside of the body for transplantation in. The last bit I want to talk about is some of the, not my work, but a colleague of mine, Professor Danny Kelly, who's using this fundamental information with advanced 3D bioprinting technology to try and regrow whole tissues or whole joints. And I'm not going to get into the details of 3D printing technology because we have one of the world experts speaking next, Gordon Wallace, at least I hope he's next. And basically how this works is you can get, in a nutshell, 3D printing allows you to have very spatial control over the deposition of cells, of materials, of regulatory factors like growth factors in a layer by layer fashion. So you can build a three-dimensional construct that mimics the native architecture of human tissue. And this is enormously powerful because if we look at some of the work from Danny Kelly's group, he's trying to grow a whole bone. So this is a vertebral body, this is a bone in your spine. He was able to 3D print a construct, so the PCL is a hard plastic that gives it mechanical support, and then he prints bioinks in between those PCL fibers, which are filled with stem cells and cues that turn those stem cells into bone. And then when we put that under the skin, we find that over time that matures into a living functional bone organ that has living blood vessels in it. So this just shows the potential of this type of technology. And he's pushing this even further because you can do a lot with this type of technology. For example, one of the main diseases we face in musculoskeletal area is osteoarthritis. Your cartilage degrades away, your bone degrades away, and the current treatment for that is basically cutting off the knee and replacing it with a large lump of metal that becomes loose after about 15, 20 years. But what about if we could print a living implant? And that's where Danny Kelly is at right now in terms of he can use this 3D printing technology where you can have a strong implant, where you can engineer it spatially so that at the top of the implant, the stem cells turn into cartilage cells, and underneath that, we can have stem cells that turn into bone cells. And this is currently in preclinical evaluation. So this all sounds amazing. But what's the ethical implications of this type of research? Well, a lot of them are good. if we think about the long-term impact of 3D printing or bioprinting, the idea, we see this cartoon a lot if you Google bioprinting, is somebody's printing a functional complex heart. We're not there yet. But the hope is that in the long term, we will be able to 3D print living human organs. And that will relieve the pressure we have on donor shortage. It's also important when you think about young people. So if a newborn baby needs a heart valve replacement, you can't put in a piece of metal or a plastic implant because that child's going to grow, the implant is not. What we want is living implants that grow with the patient. And so it has huge advantage there. It also avoids the animal question. And what I mean by that is there's a lot of significant advances being made in gene editing with CRISPR-Cas9, where essentially scientists can now make animals like pigs that essentially have human organs, which can be used for a transplantation. There's obviously ethical issues here that human bioprinting would overcome. The short term, though, where are we? Well, it can potentially avoid animal testing. If we can print human living tissue, we can use those human tissues to look at drug testing and toxicity testing, and that's some of the projects we're working on here in Trinity with myself and Danny Kelly, printing human vascularized bone. And also we can do some tissue engineering, which I've showed you already. The challenge is then social stratification. This type of technology, any type of new technology, is not cheap. So if we actually get to the point that we can do this, it is likely only the super rich can afford it. So it's not unlike the plot of the movie Elysium, and if you haven't seen it, I wouldn't recommend it. Also, we need to manage expectations. It's like the cell therapies. The potential is enormous, but we're not there yet. And so we need to kind of calm everybody down, do the science correctly, do the clinical evaluation correctly, and hopefully everything will turn out okay. Because if we don't manage expectations, we get people putting in or implanting these bioprinted tissues that haven't been tested properly, and we result in a situation like Jim Gass with the cell therapy, and we could potentially result in a black market of bioprinted organs. Another thing to think about is the ownership of these. Okay, a company is printing these, but they're printing them using your biological tissue. So you've essentially made that organ. So is it yours? Is it the company's? Is it the clinicians? Open questions. And also, how do we regulate this? I guess that'll depend. Is it a medical device? Is it a cell therapy? Is it a biologic? These are open questions that need discussion. I guess with that, I will leave you, acknowledge my research team, Danny Kelly's research team, for allowing me to showcase some of his work, and of course, my funding bodies. And thank you very much for listening.
I might just grab that and stand over here out of the way. Well, first of all, thank you to the Australian Embassy and the Science Gallery for organising this event tonight. I think all of us here really believe that events like this are critically important, and they are critically important in this area because, as you've just heard, the technology is advancing incredibly quickly. And so it's important that the discussion around the non-technical issues associated with these technologies are also addressed in parallel. I come from Wollongong, and you see it looks a little bit like Howth these days. I've never seen Howth look like that. When they dropped me off on Monday, I thought they dropped me off in the wrong country. The sun was shining so brightly and the wind was just blowing nicely. But I'm in Ireland, I believe I am now. So I work at an ARC Centre of Excellence, and let me tell you why we got into 3D bioprinting a number of years ago. We and others realised that there'd been tremendous advances in development of new materials for medical applications, for controlling biological interactions. We also realised that in order to really get the benefit out of those advances, we had to think about the way to create structures and devices that could be tested on the bench and eventually might be realised as true medical devices for clinical applications. So I just mentioned two areas of materials where there's been amazing advances in just the last few years. One is the discovery of graphene. This is simply carbon, a single sheet of carbon. You can't imagine a more boring material than carbon. But when you get down into the nano world, it takes on incredible properties, amazing strength, stronger than steel, yet very flexible, can be rendered soft as well, rendered biocompatible, and very high electrical conductivity. So if you take those nano components and you can assemble them, if you can make devices out of them, you can start to communicate with biology in extraordinary ways. You can make implants that go into the brain that give signal-to-noise ratios of brain activity that are unprecedented by a long way compared to using traditional metal electrodes or metal oxide electrodes. And as well as recording, you can use these very soft yet strong electrodes to stimulate nerve growth, to stimulate nerves so as you can communicate with them, for example, as in the cochlear ear implant, or to stimulate nerve growth for regeneration or for reconnection of nerves, and similarly with muscle. So just one example of one material that's really opened up our ability to communicate with biological systems. And I'll give an example of one other material, and it's not much more complicated than carbon. It's got a couple of other elements associated with it, a bit of nitrogen, a bit of hydrogen, but it's still an organic material and it conducts electricity. And it has this extraordinary ability to communicate with living cells that we can't get with any other material. And I'll just give one example of that. It's a recent study working with a colleague of mine who's been looking at the development of schizophrenia for 3 decades and looking at pharmaceutical ways to reverse the effects of schizophrenia at a cellular level, and that is in order to make neurites grow back out of cells where they've retracted, to reconnect branches of neurites where they failed. And they've never been able to do it with the pharmaceutical approach. So we were able to take these materials based on decades of work we've done with other neural systems and show that we could stimulate those schizophrenic cells and get them to go back to normal behavior. So this hasn't been possible with pharmaceuticals, and so the emerging area of electroceuticals using these new materials starts to make things like that possible. So you can see why materials science would be scientists around the world would be so keen to work with clinicians and biologists to translate these amazing fundamental findings into structures and devices for clinical applications. And if you look at some of those applications, all of them require some sort of device. All of them require our ability to encapsulate that new material with other materials in order to realise the true potential of the device that we might want to use, whether it's for a cochlear ear implant or whether it's for implants into the brain, for schizophrenia or for epilepsy detection and control, or for conduits for nerve and muscle repair. And on that nerve and muscle repair, just look at the complexity of these structures. Imagine you've got a damaged nerve and you need it to grow back over 5 centimetres. You need to put a conduit in there to encourage that growth, and it contains those electrical materials that can stimulate the growth, drugs, growth factors, and even living cells, even stem cells into that conduit so that you can help facilitate the regeneration of those damaged nerves or damaged muscles. So 10, 15 years ago, that was all done by hand. We individually made each of those components and hand-assembled those structures in order to just make one test device. Just make one test device. It was a very inefficient way to do research, never mind inefficiency in translating that research. But 3D printing that you've already heard about just revolutionises how we think about making things. Those new materials and mountains, stockpiles of those new materials are not amenable to fabrication using traditional manufacturing. So 3D printing turns traditional manufacturing totally on its head. As you've heard, we build things up layer by layer. We can introduce different materials in different inks when we want them during the fabrication process. And so we can create things that we could not create before the advent of 3D printing. And we can do that in a customized way to make those structures for experimental purposes or customize it for individuals in terms of the treatment that they might get. The development of the hardware is important, but so also is the development of the inks. And I'll touch on the need to have that integrated development happening in parallel alongside the driver, which is really the clinical application. We've now developed a range of inks that are capable of printing with living cells. Many of them are naturally occurring materials, but there are also some synthetic materials. And we choose the different type of ink for different types of cells. You know, whether it's cells that you want to turn into cartilage or bone or nerve or muscle, we will pick different types of ink formulations for that. But it's important that we start to identify the primary source of those materials for bioinks, and that's a movement that's really happening around the world. There's whole industries in this opportunity. in developing the primary source of the materials to make bioinks in a way that can be easily regulated and can be properly controlled to realise full translation. And when you can print those bioinks, print those biogels, those soft materials that still have enough mechanical integrity to hold that structure, even just printing the material, as you've already heard, can have some really exciting applications. So the regeneration of cartilage from stem cells, and these are, in this particular application, cells that are taken from a fat pad behind the knee and then separated and injected into that defect. Now that was a recognised therapy, but not a very efficient one, because it was basically injecting those cells into a void in the knee. Sometimes it worked, and sometimes it didn't. So our clinical collaborator, Professor Peter Chung, who's an orthopaedic surgeon, approached us with the idea of making a particular a scaffold with these biogels that would sustain the growth of those cells and ensure their differentiation into the right type of cartilage. And we were able to demonstrate that in studies with Peter. Peter's next demand was that we wanted to print the actual individual cells within the structure, not add those cells afterwards. And so we developed another formulation of an ink which would protect those precious stem cells during the printing process, maintain them in the ink in the reservoir first, and then protect them as they were put under some pretty significant shear forces during the printing, and then protect them afterwards in the right environment, that they would not only survive, but they would proliferate and differentiate into the right type of cell. Now that required the development of the ink. It also turned out it required development of fairly specialised hardware as well, because in order to truly protect the cells, we found that we had to build a coaxial printer, one that would allow the cells to come down the middle of the print structure and at the same time would surround those cells with another polymer that would protect it, another soft material that protects it during the print. And having developed that particular system, that coaxial system, we found that we could have better survival of the cells and better performance after printing from those particular cells. Professor Peter Chung, however, is rather, well, most surgeons are pretty demanding, but so Peter is no exception. The next requirement from our clinical collaborator was that he thought it would be much better if he were to print into the defect. So rather than pre-print the scaffold on the bench and then translate it into the defect, he wanted to print into the defect. So we created that pen, the BioPen. We created a structure that would allow him, or a device that would allow him to print directly into the defect using that coaxial approach. And we must acknowledge Peter's demand, because recently we've published on some large animal trials that shows that the efficacy of doing that is much better, that the ability to generate the cartilage is much better if we were able to print directly into the defect than printing beforehand and then reintroducing that into the defect. But you know, because surgeons are so demanding and mechatronic engineers can be so revengeful, that they started a renegade project on the side. And you can see that little robot there with the pen in its hand doing the printing. So, you know, if you want to get over demanding surgeons, then you build a robot that can do the surgery. But there are real, that's only a joke, by the way, that there are real applications for these robotic approaches to using 3D bioprinting. Let me return to the stem cells, because we often forget, and David highlighted this too, is that the stem cells are a critical part of the printing process. So, you know, we can print those structures and we can very quickly, and we can print the structures containing the cells very quickly. But what's not so quick is how we actually get those cells. If we're going to take those cells from the fat pad behind the knee for individual patients, we need to separate those cells from that tissue. We need to purify them. We need to make sure that they're the right types of cells before printing them back in to the individual patient. And that's very time consuming, days, weeks, in order to undergo that process. And so the whole 3D printing solution is really a 3D printing protocol. It's not just the ink, it's not just the printer, it's the cells and how we harvest them and how we get them into that ink formulation. And so we have a project at the moment that's looking at dramatically reducing that time period, getting it down to a few hours so that that whole clinical procedure can be done in one surgical operation. So, the cell component is critical, and how we address that is critical. A number of other surgeons have approached us to use that cartilage regeneration idea, this time to use the regrowth of cartilage within a structure that's framed like an ear to treat children with microtia who are born with part of that ear missing. So we've developed hybrid printing structures that enable us to get the structural integrity of the ear and the distribution of mechanical properties within the ear, and combine that with the printing of gel during the one print process, so that we can then grow the cartilage through that structural component to generate a living ear that can be implanted into those patients. Again, a project very much driven by the clinical community that we work with. Another clinical application or opportunity has arisen for us in recent years, and it was introduced to us by Dr. Toby Coates. And Toby is a clinician who treats people with diabetes by doing islet cell transplantation. So these islet cells are taken from donors and they're transplanted into people with diabetes, and these are insulin-producing cells. And again, this is a recognized therapy. And this is the immediate opportunity for 3D bioprinting, is to introduce 3D bioprinting into existing therapies where we know we can make a difference almost immediately. So Toby approached us and said, well, we lose so many of these cells during the transplantation and immediately after, because again, just like the cartilage example, it was essentially an injection process into the body. And so Toby's idea was to create 3D structures where we could print these precious islet cells, and we could surround them with other types of cells in that scaffold that would generate a vascular system to support the development of those cells, and then other cells that would help with the immune system. And so we've gone about developing the inks that can print all of those different types of cells and protect them during that printing process for 3 different types of cells. And we've just recently delivered into Toby's labs at the Royal Adelaide Hospital a prototype printer that's capable of creating these complex structures. So again, a project, an opportunity, a challenge brought by the clinical community, which required all of that knowledge about the materials, the mechatronic engineering, and the cells, and how those cells could be integrated and developed to solve that particular challenge. But there's still, even if we solve that, there's still a major limitation, and that is the number of donor cells that are available in order to treat patients. So there's a big move around the world in various projects. David talked about induced pluripotent stem cells. So it's using induced pluripotent stem cells in order to generate the types of cells that could create islets. So you can see how all of us in this bioprinting community must work very closely with those in the cell technology community to deliver results. David talked about how to figure around induced pluripotent stem cells. These are very powerful, and this approach is very powerful because it takes your cells from skin, for example, and reprograms them back to stem cells. So they're your cells, and they're back to stem cells, and so you can do then all of the things that we've talked about with stem cells using your individual cells to do that. And in doing that and in building that body of knowledge, some very interesting questions arise. Everything we've talked about so far tonight, I think, has been around how we get to practical solutions of some real challenging problems that are in front of us. And that's important, and we need to continue to do that. But I want to finish with a point that 3D bioprinting also enables us to address fundamental biological questions that we couldn't address previously. So not only do opportunities and challenges for practical applications arise from this clinical connections community that we work in, but 2 of these individuals, the individual we worked with on schizophrenia and one on epilepsy, approached us about the idea of arranging the different types of cells and starting with induced pluripotent stem cells and arranging those in such a way that they developed into different types of neurons, and we could perhaps start to get assembly of a neurally connected type of tissue on the bench and understand how those neural networks develop. And importantly, if we could then take stem cells or generate stem cells from a healthy individual versus one with epilepsy versus one with schizophrenia, start to get insights into the development of those diseases and how we might design and test treatments on the bench without animals, on the bench using the person's individual cells, test those potential interventions to see how well they might behave under those conditions. So I can report that we've taken some significant steps towards this. Just recently, we've developed an ink which is capable of printing induced pluripotent stem cells in three-dimensional structures, and we can use different conditions afterwards of treatment to develop different types of neurons and different supporting cells. So we start to move towards this idea of creating the brain on the bench through the ability to print induced pluripotent stem cells and through the ability to interrogate those cells and control their development. This has major implications. I've just gone full circle because now if we can do that on the bench, remember that graphene electrode I showed you at the start, which is an amazing electrode material. All of that information we had to get through experimentation with animals. All of those neuronal recordings through experimentation with animals. Imagine if we could create this tissue on the bench that behaved like a brain, and we were able to test our new materials on that piece of tissue that we've generated on the bench. It would save time, money, and avoid many of the ethical questions that arise through animal experimentation. And if we can do that, then we can test those materials and how well they interact with neurons or neuronal systems. Then we can come back to one of our goals, which is not only to solve those practical applications, but develop implantable electrodes that can drive wearable prosthetics, so neurally driven prosthetics. This is a 3D printed hand. It's got all of the right mechanical and flexural capabilities of a hand. It's at the moment driven by electrodes that are on the skin, but if we can develop these electrodes that are implantable, we can have truly neurally driven implantable devices. So lots of things have come together, and they've come together quickly in order to advance this area. Many areas of technology have come together, but as David alluded to, there are other things that we really need to think about. We need to be able to build strongly integrated collaborative research teams. There are regulatory matters that are very different from the traditional approach to medicine and clinical trials. And of course, all of this is needed if we're to create a system which ensures social acceptance and that the community actually wants to use the technologies that we're developing. And that aspect is very important, and in growing importance, I think, for all researchers in today's laboratories, that we do want to do fundamental research that can be translated into real applications as quickly as possible. And we're very conscious that often it's not the technical issues, but the non-technical issues that can hamper that progress, and that we need to be engaged, we need to be involved in trying to solve or at least contribute to some of those non-technical issues, as well as the progress of our science and technology. Thank you.
I'll try over here. I now know why Gordon was standing on that side. Thank you. I'm Sue Dodds. I am Dean of Arts and Social Sciences at UNSW, but I'm also a member of the Centre of Excellence for Electromaterial Science, where I lead the Ethics, Policy and Public Engagement program. And I'm going to be looking a little bit more at bionics, less in the sense of the 3D bioprinting and more in some of the more mechanical aspects. So just basic primer on bionics, so the idea of the biological and the electrical being brought together. And some of the discussion we've had so far, it really focuses on that first of the definitions. We're trying to understand the way in which we can develop systems to function like living organisms. How do we print in a 3D structure live cells? How do we develop stem cell therapies? But there are also a strong common understanding of bionics in terms of devices that can be implanted or worn to restore or repair some sort of defect or some kind of loss of function to a limb, an organ, or to a capacity. But finally, the idea that you might be able to use bionics, and for those of us who are sort of my age, which is a tiny proportion of people here, that sort of $6 million man understanding of bionics where someone has superpowers as a result of some kind of implants. So devices that can be implanted or worn to enhance functional capacities well beyond what's species typical. So, one version of that first sense is sort of the cyborg moth, trying to figure out how we could take the flight capacity of a moth and control it with a chip and with have electrical stimulation of the muscles in a moth so that we can actually get the moth go where we want it to. It's a living moth, has a chip on it, and that could be useful if you were interested in certain kinds of surveillance. The second kind may be implantable devices, or they may be wearable ones. In the case of the cochlear implant, people who have hearing impairments are able to discern spoken word as a result of using a microphone, having an electrode that then stimulates the oral nerve so that you can actually hear some kind of sound, or the bionic arm, where rather than a standard sort of static prosthetic that someone might have a prosthetic that's able, in the case of this one, to hold an egg, to be able to have a bit of haptic response, to have a bit of a sense that you can actually control it, do fine motor activity. And I think that the example of the soft robotic hands that Gordon was discussing is, again, that kind of bionic bionic device that can be used in that way. The final kind of example, and I've used a pretty graphic image from, I think, the Daily Mail, of the idea of the super soldier, that one might be able to go beyond species normal function to look at ways in which we can extend or expand human capacities. And in this case, we have the soldier of the future who's wearing an exoskeleton that bears some of the weight of his pack and his equipment, gives him also increased strength and protection because it provides him with some protection for his body, increases his endurance and allows him to run faster because it's got fantastic activity going on in all the joints, so he can spring along as if he was running along the surface of the moon. There's also a bit of the work that suggested in the soldier of the future, someone who might be able to respond to injury in the field with fast clotting blood, which is better than bleeding out, but also that it could be the case that they can control the oxygenation and the nutrients within their blood so that they can retain a level of energy and a level of strength that otherwise might be missing. And then there's the idea of also that there might be neural enhancement so that they can have telepathic communication between people and brain control of machinery. And that may all sound very far-fetched. They're not that far away. The idea that people can, through brain control, can move things in the world at a distance is certainly currently being investigated, and there's early tests showing for that. Similarly, enhanced senses, infrared vision, or capacity to hear things that otherwise might be missed, and then some of the discussion about genetic modification. And many people find the idea of moving down this sort of hyperhuman to be very concerning, and often the discussion about bionics trips a bit on that possibility. But if we think about medical bionics in the kinds of cases where we're trying to repair a defect or an impairment, there's a number of features of the development of these bionics which are exactly common to any other development of a medical technology, including concerns about safety and efficacy, both the use of animals in animal studies and concerns about doing that, but also ensuring that we've got good models for the effectiveness of, or the safety of a particular bionic device through the animal studies that we're conducting. Questions about cost, whether or not this is a good investment for society to make, especially if it's the case we've got differential access to the technology once it exists. Questions about impact on quality of life, whether or not this actually addresses the need that a person has. And for some of those kinds of implants or wearable devices where we find that you get better outcomes for people if you start very young, such as the growing of the heart valve in a child, questions about consent, or in those cases where we might wish to use some kind of bionic implant for addressing degenerative neural conditions or certain kinds of neural conditions, questions about consent of the person in those schizophrenic studies, for example. More specific to bionics, we would find questions about long-term stability. If something's going to be implanted in me, I want to know that it's going to be either stable for a long time and therefore not do me damage, or appropriately biodegradable, and when it's no longer needed, be able to work its way out of the system, and not something which is going to cause infection or damage to me along the way, or to cause any kind of environmental damage. And a question that came up certainly in the development of the cochlear implant, and often in discussion about what kinds of prosthetic devices we ought to have, is the way that development of bionics may affect our understanding about disability. If we don't listen to and understand the concerns of people who have impairments, people who have limitations, which other people think a bionic may be a response to, we may find that we've got bionic responses which are not the ones that people will actually want to use, and they may continue to perpetuate stigmatization of people who choose not to use them. So in the deaf community's rejection, in some members of the deaf community's rejection of the cochlear implant, that was one of the concerns. The use of the technology, is it being used to control populations? Neural implants that may be able to shape people's impulses sound like one way of managing people who have impulse control issues, but it may also be a way of a society controlling people who maybe seem to be difficult. The control of the technological design and what kinds of things, who gets to determine which technologies get taken up, and especially when that moves into questions about commercialization, we find within Bionics the possibility that I end up with the wrong kind of device so that I can't be upgraded at some point in the future. If this is a lifelong implant, that will have a lifelong impact. Questions of hacking, if we're using Wi-Fi or Bluetooth in order to be able to tune my neural device rather than opening me up, which I think is probably a good thing to use Wi-Fi or Bluetooth for that, there's a possibility of being hacked. And what should we do about that? Manufacturers' liabilities clearly are going to be lifelong questions, and how we as a society determine what the responsibilities and liabilities of manufacturers are will help to shape the kinds of technologies we may have available, because some manufacturers are not going to develop things if we've got a very restrictive set of responsibilities on them, and this may be one where we need as a society to talk about where does responsibility lie. Questions about upgrades, as I've mentioned before, if something is wearable, it seems less concerning to be able to have an upgrade than something that's actually inside my skull. I'm just going for the dramatic version there. And environmental impact, obviously, we've got on the one hand, if something is intended to pass out of my body, where does it go downstream? And the other is if I've had an implant, what happens to me on death? At the moment, people have discussions about what happens with pacemakers on death and so on. And then the question of transhumanism, which gets us back to our super soldier of the future. So let's just very briefly talk about those ideas of therapy and enhancement. There's a concern by some that we should not use bionics to go beyond what normal human capacities are. And there's a concern within disability studies that in framing that discussion in terms of therapy versus enhancement, we start with an assumption that there is a single set of characteristics which are common to all humans rather than a wide diversity of capacities. And as a result, we're trying to say that everyone needs to come up to the same standard, but they shouldn't go any further. But even within disability studies, there's discussion about therapeutic enhancement. The idea that, well, look, if at the moment the bionic eye is simply going to allow me to see shapes and have a bit of a sense of the edges of things, that's not going to help me to be able to distinguish between living human beings and mannequins. In order for me to be able to get through life in an effective way, it's useful to be able to distinguish humans from mannequins. Maybe infrared's helpful, because at least I'd be able to figure out which ones were more or less warm. And so then I'd be able to get through life and burn myself less often, be able to get through life with one less impediment. So if it's the case that I'm going to have a diverse range of capacities even after I've had the bionic implant, it may well be the case that we don't want to just stop at some fixed norm of human capacity. We may wish to say, well, what is it that the person can actually do or be? And is it the case that this particular technology can get me to where I want to be? And again, this is best assisted by talking to the people who we see as the intended beneficiaries. In enhancement versus transhumanism, there's on the one hand a sort of an aesthetic of transhumanism, people who wish to hack their own bodies so that they can be able to tweak their experience of the world, so that those are people who often will want to have some kind of mechanical implant in order to allow them to run their phone or their fridge from their body. But in other cases, people are saying, well, look, actually what we want out of transhumanism is the freedom to go beyond the current limitations of the human species. And that given we allow that people might perfect their athletic abilities or their aesthetic abilities in dance or whatever, we don't see that as something which we try to restrict, even though they go well beyond what otherwise would be the normal human capacity. Why should we stop people from engaging in enhancements that they wish? But in doing so, they're changing the shape of the community we're in. And we need to have that conversation about what kind of community are we developing. Briefly, in dual use, you know this one yourselves pretty well, that those ideas of the new bionic soldier clearly has to raise some questions. One, are we creating a new biological arms race, a bionic arms race? Is it the case that we're making it so we've got more reasons to be concerned about who's got the technological skill? Could soldiers be hacked? What would the effect of that be? Should we be worried about civilian life? I mean, most people in most societies, if they become soldiers, they do it for a period of time and then they stop. If it's the case that we've engaged in neural tweaking, as well as changing their sensory capacities and their strength, is it the case that we're going to cause harm? So again, we've got a range of issues that come out in each case. People assume that implants are safer that wearables are safer than implants. There are serious risks associated with implants that aren't available in wearables, but it's also the case that the shift from wearable to, oh, that's nicely done, from wearable to implant is rapidly shifting, because we now are able to have patches that actually allow for the detection of what's happening in someone's skin and being able to transmit drugs back into the body as a result. So it doesn't seem quite so clear that a wearable is completely on the outside and an implant is wholly on the inside. So I'll just end, because I know I'm a bit over time, that there's a number of ways we can work through the ethical development of bionics. Ethicists working closely with researchers and regulators, anticipating some of the regulatory gaps, finding ways to talk about the developments that are going on, especially with those people who we see as potential beneficiaries, increased transparency about side effects and about the full outcomes associated with the technology, and having a debate about what we see as the responsibilities of a society, manufacturers and individuals, in order to be able to have a regulatory and a responsibility framework that can succeed. That's the team, and that's the set of institutions associated with us. Thank you.
I think we've had three remarkable and very sort of interesting presentations tonight. So again, I'd very much like to thank David, Gordon and Sue for their presentations tonight. And we now have some sort of times for questions and answers. Perhaps, if I don't mind taking sort of the first one. Sue, you mentioned about we need to have that sort of conversation about, all right, where do we go with sort of like enhanced humans, or do we just, repair and sort of get there? So who is the who? I guess it's the question. Is it the researcher? Or do we go to the other end where it's, the person whose body that we're talking about? Or is it something in between? Because we talk about sort of, having these conversations. What's the role of government? What's the role of the researcher? And, where do we go from there?
Look, I think it's all of the above. But I think that it's trying early enough in the development of the technology to start a conversation with communities such as this one, to have experts in the room, to have regulators in discussion. Within ACES, we've regularly had conversations with the TGA, the Therapeutic Goods Administration within Australia, about things like their views about autologous stem cell research, thinking that, well, there wasn't a need for specific regulation in that area, but now we're 3D printing live human cells. Perhaps there needs to be a discussion about the therapies that would come out. The regulators don't know which way it should go. They need to be discussing with both people who are clinicians, with potential recipients to find out what's the community response to this, but also with the researchers who are developing. So I think that those conversations, and I always like to be in because I'm just a sticky beak, but I think that having those discussions, but also uncovering what people's concerns are, things that we might not have thought about. You know, what happens to the intellectual property in some of these cases? We know that with pacemakers, cardiac pacemakers, that the most difficult bit is getting the right wiring, so that if someone, if we need to repair anything, we need to know what are all the constituent parts, which we might not have thought about. We tend to focus on the working bit of the technology, not the way in which it's plugged in. Well, there's probably similar sorts of things going to happen with neural implants or with other implants as well.
Great, thanks. Now, if you'd like to sort of ask a question, if you could sort of identify who you'd like to ask and stand up and ask away. Don't be shy, I'm sure there's questions out there. Yes, up the back there. Sorry.
I have a question for the first presenter. You mentioned the organs, the printed organs that would grow with the organism. Have it actually been confirmed experimentally that it can happen?
So you're asking, has essentially a tissue engineered organ been implanted that has grown over time?
Yes.
Off the top of my head, I can't think of an example, but what we need to think of is that, okay, we're putting in, let's say, part plastic, part cells, part biological inks, but they're designed so that that plastic will degrade over the space of maybe 5 to 10 years. So that's completely gone. And over time as well, the cells are replaced with host cells that are there. So it's not like the implant is growing over 10, 20 years. The implant that we put in is completely gone after a couple of years, and what's there is a completely regenerated tissue. So I strongly believe that would work, but I can't think of an example off the top of my head. But you have to think it's not an implant after a period of time, it's just a regenerated tissue.
Gentlemen, just at the back.
I wonder what do the panel think about the global context of trying to preserve life and extend life in the context of a planet now that has 8 billion people on it, and particularly if a lot of these technologies are very high value or high cost, and how does that feed into global research priorities compared to other areas?
I'm happy to start.
Okay.
And you guys can fix it. Look, I think that's a serious question. It's a serious question about where do we want to put our effort. But given that some of the technologies we're talking about, well, they have an initial high cost, they come down quite quickly. But it is about then thinking, well, which things ought we be focusing on? I mean, islet cells in pancreases seems like a really good one, given the level of damage that diabetes causes across the world. But I think your bigger question about how do we actually ensure that these, in principle, technologies can be then extended so that they can have bigger health impact around the world on those conditions that affect people in developing countries. I regularly tell Gordon that my fantasy is that we would be able to use some of the microfluidic diagnostics to be able to test, say, for different strains of malaria in different very distant parts. I'm at Papua New Guinea and up in the mountains, and being able to use our technology to then print out time-released drugs that would be delivered through the skin, so you don't need the usual travel into an urban centre in order to get the treatment you need, and you'd have a cheap, effective way of addressing malaria. That would be my fantasy. If we can do that, I'll feel like a happy woman.
Yeah, if I can just go onto that particular application, but just thinking about that question, I guess if you think about all of the things we're doing in 3D printing and bionics and tissue engineering at the moment, it's all about improving the quality of life rather than extending life. And I think there are other things that do extend life, and but there's obviously a big opportunity across the board from younger, middle-aged to older people to improve that quality of life through these advances. And I don't think that's why they've formally been chosen as projects, but I think in retrospect, if you look at it, that's really what the emphasis is.
I think I might just add a nice example of what we have here in Trinity, kind of related to what Sue was saying there. If you think about the third world, their access to medical devices is pretty limited. And if you did send them a medical device, if it breaks, they're in trouble. They're not going to get a new one. And so what we can do instead is we can send them a 3D printer, which not a bioprinter, it's a standard printer that prints plastic. It costs about €2,000. And what we can do is we can send that and then send them software files that allows them to print medical devices when they need it, like casts or tweezers or these little things, or even stethoscopes, and they work. and that's a student-led program here in Trinity, where the students are designing these medical devices that can just be sent via an email to anywhere in the third world and then printed.
Yeah, and I think eventually, and probably not in the too distant future, that bioprinting will become customised enough for particular clinical applications that same concept could work. And it's certainly our intention to do that, for example, in the 3D printed ears project and in India, for example. And another is one we didn't talk about tonight, but the iFix pen for corneal regenerations or damaged ulcers in the eye. Again, there is a field version of that which is being developed specifically for that clinical application, which is simple to use, easy to transport, and easy to implement.
Any further questions?
My question's for the first presenter. You briefly spoke about conditioning stem cells before injecting them. Is that currently being looked at, or how might one go about doing that?
Yes, it is, I suppose. So when we take stem cells out, they're kind of a very pure population, and we can expand them in certain growth factors that kind of fine-tunes or selects a certain population within that stem cell population, which can tend to be more regenerative. And I think that's really, really important because, we have a study in my lab ongoing at the moment where we're trying to compare stem cells from young, healthy people that we've extracted. So these are 20-year-olds that have, donated their bone marrow. And then we're looking at stem cells that we've taken from 85-year-old osteoporotic women. And the general thinking would be, their stem cells are poor and they'd be really bad at regenerating tissues. But we find that's not the case at all. There's such variability in stem cell populations across donors that we find that we have a healthy 20-year-old, and when we try and get that stem cell to make bone, it's really poor. But then we have this 85-year-old woman who's suffering from osteoporosis, and her stem cells are amazing. So if I want a stem cell therapy, I want it from the 85-year-old. And I think in terms of conditioning, maybe rather than just taking stem cells and putting them in, we need to figure out what makes a stem cell a good stem cell and condition it that way and then inject that in.
Right at the back row.
Yeah, hello. I have kind of a vested interest. My daughter has epilepsy. She's going to be 20 next month. And I'm just wondering, I know it's all fantasy land at the moment, but how far ahead do you think they might have effective treatments for epilepsy with these graphene electrodes, et cetera, et cetera? And were there many trials done yet at all? I'm probably not. Just curious a little bit more specifics about that, please.
So I'll start that off. I think the sort of fundamental breakthroughs that we're looking for are some years off in terms of revolutionary new treatments. But of course, along that journey in developing better implantable electrodes, for example, that can be implanted into or onto the brain, there are projects that are well developed in terms of using these improved electrodes to warn of the onset of an epileptic seizure, so as at least the individual knows that the event is going to occur. That in itself is a big step forward. And there are other projects that are looking at taking that information and automatically responding to it either chemically or electrically in order to circumvent that seizure occurring. So as we look for more revolutionary new treatments because of the advances in the technology, I think there are incremental advances. There are still very significant advances that will benefit patients or individuals with epilepsy.
My question is for presenters 1 and 2. I was wondering, in working with the stem cell grafts, do you find that, be it in vivo or in vitro, do you find that in going through the step of inducing them to differentiate and proliferate, that neoplasias are a common side effect, or does that unrestricted growth not happen as often as one would imagine?
Yeah, in vitro we don't see any issues there. In vivo, if you look at cell therapies, those original stem cells seem to be very, very safe, from at least what we're seeing from the clinical trials. And one of the reasons is that they're washed away after about a week or two. So they get to the site of injury, they release these factors that regulates the immune system and trophic factors to promote blood supply and so on. And if you go in about a week or two later, you can't find them. And so they don't have long-term adverse effects like that, at least in adult stem cells.
Yeah, and I think we've seen just positive effects in vitro. There hasn't been a lot of in vivo work done. There's an increasing amount very quickly in vivo in animals, at least, with the 3D printed stem cells, but it's early days yet.
Hi, just a question about the exciting vision that we see here. Over the last 5-10 years, I think the advances have been astonishing, but this has required the mobilization of a research effort across many disciplines that you normally don't see you don't see it in universities, for example, not very easily. It doesn't happen easily because universities are not organized that way. So I'm just wondering about how this research effort, which has got such tremendous potential, how can it be organized, mobilized, sustained, and directed towards trying to solve these big problems, while at the same time being careful about the conflict or tension that might arise within the traditional disciplines in the university, because that's where the feed-through of knowledge comes from.
I can start that. I think that the drive at the moment is, and the term which is very appropriate, I think, is that this is anti-disciplinary research. And so what we're looking to do is to break down those traditional barriers. But I don't think you do it by telling people we should break down the traditional barriers. I think we do it by demonstrating the potential impact of doing the research in an integrated anti-disciplinary way. And that impact needs to be explained to all involved. The researchers as well as the funders and the university administrators will eventually catch up with it. So let's charge on and make sure people understand the vision, the benefits that this approach can deliver, and that will get people excited about being involved in it and get people more flexible in making sure that we do support it. That's what I think we need to do.
I think here in Ireland, obviously Science Foundation Ireland have initiated this research centre programme, and that essentially forces disciplines to come together to form large collections of researchers from different backgrounds. And I think we're here within the Science Gallery, which is Amber, which is an SFI centre which is based above it, which is Advanced Materials and Bioengineering Research, and that brought together biomedical engineers, physicists, chemists, and a lot of the bioprinting work is being done as part of that Amber Research Centre.
And I think and obviously that collaboration is more than just across the technical disciplines. As we spoke tonight, all of us, I think it's collaborating with the community and the end users, collaborating with the regulators, collaborating with industry. It's really making sure that we have all of those elements that are needed for success, and very importantly, the clinical community. All of those elements needed for success engaged in the work as early as possible and engaged in the journey.
And I think that one of the things it does is it helps to address some of the frustration that people feel about why don't people just take up this idea? Why can't this actually become effective? And if you don't have the complex capacity to address the problem, then you only get partial solutions. And so that sort of, as Gordon calls it, antidisciplinary, I kind of probably wouldn't use that term, but there you go. But that capacity to say, well, I'm not bounded by my discipline when I'm trying to solve a problem with others, that you actually can get to a point of resolving major issues, not just the bit that you know you can control. And I think that increasingly the kinds of challenges we're facing in the world require that respect between the different backgrounds to be able to address those challenges.
Yes, just over there.
Yeah, I was just wondering about, do you think it's perhaps a problem if in the future, say, those like countries and places perhaps which don't much care about the ethics of things or don't have much of a regulatory framework, they might sort of overtake those places who are trying to do it in a more cautious and responsible way, let's say?
Look, my perspective is that absolutely there will be things going on that don't meet regulatory standards of other countries. But it's also the case that this is where publishers, manufacturers, and others have an interest in trying to ensure that what ends up on the market is of high quality. That publishers have a responsibility for knowing whether under what conditions did this research occur, and it's they've been reasonably good at refusing to publish things that they thought, well, hang on, this wouldn't get past an ethics committee elsewhere. But also manufacturers have a very strong interest in being able to demonstrate that the products they've got have been tested in good conditions, that they've been gone through a good process of regulation, because if it's going to be expensive, people want to know that it's good quality. Now, that's not going to cover everything, but that international pressure often will come from saying, well, one, how do the scientists engage with each other in terms of the quality of the research, but also how do organizations, how do healthcare systems respond to people who may, as a matter of healthcare tourism, have had a treatment somewhere else and then come back and expect to be treated for complications that have arisen from something that happened while they were overseas. So questions about health insurance also need to be brought in this. And this is a reason for saying that with those ethical and social impacts, we need to think about those early on and think about them broadly. Often societies will say, well, we'll have really, really tight regulation. But if that means that the population are going off and getting whatever the treatment is that's not as good quality from elsewhere, then you've got another kind of problem. So those need to be thought through really early on, rather than wait until you've got a problem. Yeah.
Just a sort of a question. I've seen sort of, I guess, the wonderful advances that sort of stem cells have sort of done. We had a family member who has MS and travelled to Italy and had sort of stem cell sort of treatment, and it really sort of halted the destructive sort of path of MS and improved her quality of life. But one of the issues was, she was very lucky in that she had some relatives who were wealthy and offered that sort of treatment, and she was able to sort of avail herself of it. But these sort of wonderful sort of advances, they cost money. They're very expensive. And often the people, are we going down the path where it's going to only be the very wealthy? And perhaps, Gordon, is 3D printing offering sort of the ability to, I guess, reduce the cost? Does it make it sort of more accessible to people?
Most of the clinicians that we work with, that is certainly their point of view, that what they're doing is producing. And of course, a lot of the things that we're working on are not solvable by traditional means, so it's hard to do that relative comparison. But if you look at some of the other projects, like the iFix pen, for example, with 3D printed ears, the idea is that you are actually producing something which costs less than what's available at the moment. And that's a question it is addressed early on, as it should be in most of the clinical work that we're looking at.
One more question?
Okay, the lucky person up the back.
Hello, I'm just wondering, do you see any immune reaction to these implants in either animal models or in humans?
Yeah, I'll start it. Yes, as with any implant, it's important that that's taken into consideration, and there will be some response. There are some interesting strategies that can be used through 3D printing to try and control that immune response, so it certainly can't be ignored. It'll be the same as with any other implant, but there are some aspects of the arrangement of materials and other biological entities within 3D printed structures that can hopefully help in controlling that.
Yeah, I mean, here in Trinity, there's a lot of work in that type of area. I mean, if you look at tissue engineering over the last 10 years, a lot of the in vitro work, or the work done in the lab, is you take a material, you stick stem cells on it, and you get bone. And you think, oh, this is fantastic, I'll stick this scaffold into the body, stem cells will come into it, and hopefully you get bone, right? But what we didn't think about is actually the first cell that touches that material is macrophages, it's the immune system. And it's those cells that then in turn react to that material and then send out signals that start regulating that stem cell behavior. And so now the tissue engineering community is really beginning to think about the impact of putting these materials into the body in terms of how macrophages are reacting with it. And we see huge effects on just even the morphology of the material, the stiffness of the material. All of that affects the macrophage behavior, which in turn affects how they control stem cells coming in. So it's a good question, and we're working a lot on it.
Well, that concludes our sort of presentations tonight, and I hope you join with me in thanking David, Gordon and Sue for their wonderful sort of presentations and their time taking in answering our questions this evening. Thank you. Now that does